Okay, thank you. Hi, everyone. So I will briefly present ongoing work and future perspectives of this working group. If you recall, the main aim of the Workgroup 6 database was to evaluate background levels and sensitivity to the assay using the data from controls. At the time, we thought that given the broad expertise of the working group members, it would be interesting to gather information also from in vitro cells and animal cells, besides the human biomonitoring. And for each of these models, it was asked specific data related to the cells that were used and the processing methods of the cells, as well as technical issues related to Comet, including enzyme information. The greatest contributions were for human-based models. And with the data provided, we confirmed the preference of whole blood and PBMCs for Comet evaluation in both human biomonitoring or in vitro experiments using human cell lines. It was also possible to find that for assessing the levels of oxidative DNA damage, FPG was the enzyme used, and preferably of commercial origin. So, as decided in the last meeting in Turkey, we asked the help of Stefano to see what could be done in terms of statistical analysis. So we concluded that there was an overlap between our data and hCOMET database that had already some important conclusions, as you've seen. And regarding the animal cells, there were high variability of species and types. So we decided to try to realize the potential of for meta-analysis using the data collected from the in vitro studies. For that, the data had to be edited, focusing on cells with more entries. We also have to diminish fields, although maintaining the ones that can be variable. Sorry, are you hearing me? Hi?
Yes,
yes.
Oh, and because I didn't hear you, sorry. And the data was added, and we will see hopefully in the next weeks what can be done. You see here is how the editing was of all the data. We have only 3 cells for cell information, 4 or 5 for technical information and enzyme. So we will see how this works in a meta-analysis in the next weeks. During these last 2 meetings, it was also discussed the possibility to do a literature review on human cell lines, but by organ type. The idea was to do a systematic review. The template and the protocol is already a draft, and also considered and draft is a quality score sheet to score each of the paper retrieved according to a number of criteria that it's believed to be important to see the quality of a paper. So the tools on this are more or less done. So we need just workforce to put this in perspective. The last is the cell inventory that would be of great help either for beginners and also for the experienced people researchers on Comet, because they can always want to change to start working with other models. So the main aim is to make it available on the hCOMET website, as already have been said, and 2 templates were proposed, and this one seems to be the winner, as it should, since it was Andrew's table. And so this is it. Thank you for your attention, and I would just have just a few words. I want to say that I've enjoyed a lot to be part of this cost action. I've learned a lot with you all, and I hope to continue the work. So all those said, I'm excited for the next action and to see regarding Comet. I hope you continue this work. Thank you.
The Workgroup 6 was already presented by Solange, the most important work, which was database. And this group deals with comparative study of different cell types, led by Solange and myself, next one. And it was a really long journey from 2016. Andrew, can you move the next? And you can see how many meetings, physical meetings we had, basically 2 per year. And it was really excellent experience and lots of lively discussions and ideas. And I think this strengthened our network. So the final meeting actually couldn't be in Budapest or Croatia, but it's here. But last time physically we met in Antalya in October 2019. Next one, please. So the objective of our working group is the fits to objective 7 in the cost action to assess the suitability of different human cells, cell types for Comet assay analysis. And we set up our tasks ourselves, and most of actually outcome you already heard. So it was the combined database where we had an information about the cell types, primary cells, human cells, cell lines in vitro, but also in vivo. And this was great work of Solange. Thanks really, Solange, for this. And I'm sure this will be also useful in future. Then we were supposed, and we did, we assessed suitability of frozen whole blood cells as a source of leukocytes, and this was done by many STSMs and also by individual laboratories. So we have lots of outcomes from this exercise. Then we were supposed to do protocol for isolation of the cells, peripheral blood mononuclear cells from blood samples, and this actually activity is aligned with working group 4, which is supposed to do SOPs, and this paper actually under supervision of Peter Muller is now under preparation, and I'm very happy for that. And actually I must say that all our activities are aligned with other basically with all working groups, because you can't do Comet assay without cells. And so this last task was to explore feasibility of using stored Buffy code as a source of leukocytes, and this is aligned with working group 7 and others. And Buffy code samples from the German EPIC cohort were already reported by Eilul from Helga's Stopper's group, and I'm really very happy for this outcome. Next one. So the milestones, we had the milestones to recruit of cost members with mostly own resources to take part in the working group 6 tasks. And of course, this was through meetings, STMS and trainings and all the discussions and networking we did. And I think we reached these milestones absolutely more than 100%. And we set up our own deliverables, which was the report on comparative data retrieved from combined database. And this is, of course, in collaboration with Stefano and Mirta, working group 1. And it's ongoing activities. We hope that until the report is due, which is in months or 6 weeks, we will have results on this. And then the next deliverable is recommendation of on suitability of different cell types for the Comet assay, and this was addressed in special issue we actually initiated, and also as Solange already mentioned, we are planning to produce the catalog of cells. This is under preparation, and it could be either on website of hCOMET or independent database online as a live database which could be updated. And then we had a task to and deliverable to produce publication on level of DNA damage in leukocytes and peripheral blood mononuclear cells. And there were several papers published, and one is already also on preparation. Next one, please. So this is actually kinds of demonstration that the most valuable or most used samples are peripheral blood mononuclear cells, but there are also other sources of cells such as buccal, whole blood, buffy coats, and saliva. And the biggest potential, as you could also see today from Sabine's and the Eylul presentation, that buffy coats is actually great potential to do retrospective studies. So I will not spend time on this. Next one. And there were overall more than 30, actually, I counted, maybe JP can correct me, 34 short-term scientific missions, and many of them, if not all, were related to Working Group 6, because they of course you need the material cells, and this was a really great achievement. The next one, I will not mention all of them, and I asked all Working Group members for sending me slides, and I got a lot of nice presentation and slides, and I can't present all. Just two as an example. This is STMS from Vienna. Francisca sent me yesterday very nice presentation, and it was mission to Sabine's lab, also related to repair. But you could see here the blood collection from four different types of surgery and 3 times points before the surgery, months after surgery, and 6 months after surgery, collecting bloods. And on the left side, so you can see how baseline level of DNA damage decreased after 6 months, and also the same was with FPG sensitive sites. Next one. And then it's also very, very lively and long-lasting collaboration of Karina and Lisa, and they actually compared different types of cells, whole bloods, buffy coats, and isolated mononuclear, peripheral mononuclear blood cells. And you could see here that the baseline level of DNA strand breaks, as well as FPG sites, net FPG sites, are actually very similar, or I could say the same for whole blood buffy coat and peripheral blood mononuclear cells. Of course, it's different for H2A2 hydrogen peroxide induced breaks due to the known fact that whole blood cells that there is no response, and the same similar is also in buffy coat, but very strong response in peripheral blood mononuclear cells. Next one. There were lots of publications, and as an example, it's here just Karina's and Lisa's publication for cryopreservation of DNA damage in different blood cells preparations. And also I would also like to highlight Goran's 2 papers on animal models, because this was kinds of parallel outcome from the database Solange was responsible for. So next one. Our working group actually initiated 2 special issues, and of course it was in collaboration with other working groups, and the special issue 1 was in collaboration with working group 1, Stefano and Mirta, and Andrew was guest editor, managing guest editor, and 3 of us were supporting guest editor as well. And this actually was the outcome of 15 manuscripts, and 10 of them with international teams of co-authors. And the next one. And a second special issue was focused on COMET assay in vitro, because we early realized that even this is focused on human biomonitoring. The most of the labs, if not all, are also dealing with in vitro COMET assay using primary cells and cell lines. So this special issue was the outcome of this was 20 manuscripts, and all of them were written by hCOMET members or observers. The next one. So the next slide shows just an example from the special issue 1 on human biomonitoring, but the next one is the whole list of whole list of special issue manuscripts of special issue 2 on in vitro. Can you move it? There were several again as well, Andrew. There were several groups of papers. Can you press, Andrew, again? So first group was dedicated to technical developments. The second green one was dedicated to different cell types, and these 8 papers actually will be source of the catalog, because these 8 papers address the different cells from different organs origin, and the third one, blue ones, are dedicated to genotoxicity, and last one, Andrew, this yellow 4 papers, anti-genotoxicity or on and antioxidant protections. So it was quite a good collection of papers. So we have still ongoing work beyond the hCOMET action, and the main is the database analysis, and the second, the innovative catalog of cells, which we hope will be live document, something similar to wiki or gene sequencing, that it could be updated with the useful information about usefulness of the cell lines and also the protocols. There are several publications under preparation, and many of other activities are ongoing. And one of them, which I really hope we will continue, is to promote COMET assay for the test guidelines, OECD TGs, because we know it's in vivo COMET assay, but still not enzyme modified COMET assay, and it would be really good to have in vitro test guidelines, and also test guidelines on human biomonitoring. The next one. So for me, it was the great experience and lots of good outcomes, but the biggest impact is that we together created a huge COMET family, and I would really like to thank to all of you who were so active and collaborative, and we built lots of friendships, and I think this will survive beyond this human hCOMET cost action. So thank you very much. Yeah, I am also impressed with the Buffy code results. This is just like we say, the cherry on the top of the tart. So it's so promising that all stored samples could be used, or many of the studies which have a samples of Buffy code could be used for retrospective studies, and also for future the proposals, because I don't think we should stop here. And I was looking today on my history, and actually before the COMET, there was in Croydon and Prague, and also then in Warsaw, we built the network of human biomonitoring COMET. So it was 20 years ago where we had this idea. So I'm so pleased that it goes from generation to generation and will continue.
Thank you, Maria. And there are lots of nice comments. I hope they'll be preserved.